Hello, everyone. I am Leah Schust, the co-founder and president of the FamilieSCN2A Foundation, and I have the honor to start off this incredible meeting today. We are thrilled to see such an amazing turnout for our first ever virtual conference. I think we heard that there was at least 100 of you joining. So thank you very much for taking the time to listen in. There are two big reasons that we're hosting a virtual conference. One, this is our off year from our biannual family and professional meeting, which is an in-person conference. And two, we understand that traveling is not feasible for everyone, especially with our children's needs, and we want to reach as many of you as possible. Our hope with this conference is to share information and education with everyone. Excuse my interruption there. I'm having a little bit of trouble moving my slides. Lindsay, thank you. So our hope with this conference is to share information and education with everyone interested in SCN2A, including parents, caregivers, clinicians, researchers, and community members. The great news is there's just too much information to share in just one sitting, so we will be breaking the content into two separate conferences, both accessible virtually. Can someone advance my slide, please? Thank you. Today we will learn about the progress made in basic science with understanding the SCN2A gene, its function, and what happens when there is a change. This type of research has placed SCN2A on the road to new treatment discoveries. Also, in conjunction with understanding the function of the gene comes the ability to define the clinical presentation. We will review the spectrum, symptoms, and get in depth with a couple of the most common comorbidities. Part 2 will be held on November 7th and will center around therapies that have been shown effective for helping our children live a better quality of life while awaiting new treatment. Could you please advance the slide? With the recent influx of new families joining our group, I thought it would be helpful to go over who we are, what we do, and how we do it, as well as the why behind it all. The FamilieSCN2A Foundation is an all-volunteer-run organization created by parents of children diagnosed with epilepsy and/or autism as a result of a change in the SCN2A gene. Our vision is to find effective treatments and a cure for SCN2A disorders. Our mission is to improve the lives of those affected by SCN2A disorders through clinical research, effective treatments, public awareness, early detection, patient advocacy, and family support. Advance the slide, please. This slide shows our roles as board members. The photo in the bottom right was taken at last year's Family and Professional Conference, and we've grown since then. If you would like to learn more about each of the board members, please visit our website and click on the About tab. As you can see here, please advance the slide. Each board member also chairs a committee, so that we have community support, finance, fundraising, international outreach, marketing and PR, and of course research. And several community members have stepped up to serve as well. You may also notice that there are several open positions on these committees. We would love your help. Please consider joining us in the fight to find a cure. Next slide, thank you. When our son was diagnosed, we were told that SCN2A was so rare that they could not connect us with another family, let alone a community. This, as too many of you know, is almost worse than the actual diagnosis. And I started this Facebook group with the simple goal of finding another mom to commiserate with. As the Facebook community grew, a few of us took it to the next level and started the 501c3, and this led to a successful international medical foundation supporting over 350 families from 23 different countries. We met our first goal pretty early on, and that was just to get SCN2A on the map. And since then, the awareness has grown even beyond our expectations. We will not stop until no family is told at diagnosis they are the only ones. Next slide. These faces here represent the why. This is why we fight so hard and the reasons behind every single thing we do. Next. And here are some of the precious lives that we've lost to SCN2A, and this is a reminder that we need to fight harder and faster. Last slide. Last and most importantly is how did the Families SCN2A Foundation and the researchers studying SCN2A make so much progress in such a short amount of time? The key word is data. We joined forces with Simons VIP in 2015 to host our most valued possessions, your data, our family's data. The options were overflowing, and we were strategic in choosing Simons VIP over others as we had one big goal in mind, and that was to make it the easiest process for families and the most beneficial to the greater research community. Simons was able to offer all of that and more. You'll hear a lot more today about how the data has been collected and used to define the disorder. We're so very grateful to Simons for the opportunity to leverage ourselves with a robust longitudinal registry. So I will stop here and thank everyone again for joining, and I hope you enjoy the conference.
All right, thank you, Leah. Our second presentation today is going to be Dr. Wendy Chung. Dr. Chung is a clinical and molecular geneticist. She's the Kennedy Family Professor of Pediatrics and Medicine at the University of Columbia, and she's one of the principal investigators for Simons VIP. Take it away, Wendy.
Thanks, Lindsay, and thanks, Leah. So I'm going to go relatively quickly through this, but as Leah was saying, all the data that I'm going to be showing is from individuals who are in Simons VIP, and this is part of our ongoing commitment to be able to give the data back to the community and help you understand it better. Just very briefly, in terms, I think many of you, or maybe all of you, hopefully know that the genetic things that I'm going to talk about, for the most part, are things that happen new in your child, or what we call de novo changes. They are genetic, but they're not something necessarily inherited from either one of the parents. As a medical geneticist, many people ask me, where do these things come from? And I just want to reassure the community, many of you have heard me say this before, it's not anything that you did or didn't do during the pregnancy that caused this, but none of us is perfect, and these genetic things happen in every single child we have. However, in your child or family member, it just happened to be in the SCN2A gene. So I want to give you an update in terms of the information that we know about and that I'm going to talk about. The first is that there are 114 individuals registered so far for Simons VIP. It's not a requirement, but of those individuals, some people have consented to be part of the registry, and so it's 86 of those individuals have consented and have submitted their lab reports. The reason that we do that is we want to be sure that everyone is in the right club and that this information is in fact correct, so we double check it to make sure that we have it recorded correctly. Of those, and this number will change over time, but of those 86, 62 were able to be verified as being either what we call pathogenic or likely pathogenic. So in other words, that we think the SCN2A is the answer. You're going to hear more information about sometimes how we can understand whether or not these genetic changes in fact affect the way this particular channel, SCN2A works, the sodium channel works. But of those 62, 52 of you then went on to do an interview with Ashley Wilson, one of my genetic counselors, to provide medical history information as well as developmental history information, and that's the data that I'm going to be sharing with you today. So I know this is overwhelming. It's not meant to be something that you digest, but this is a list of all the different mutations that we're going to be talking about today. Individuals have these different mutations. This is not as much so much for the families as it is for the researchers, because they're interested in being able to study many of these different mutations. But the big thing I want to point out is that you'll see in each of these a column of the number of individuals, and with one or two exceptions, you'll notice that the number is 1. That is that almost every individual is the only one so far with the particular change that we see in this channel, And therefore, to me, what that means is it's really important for all the individuals to actually be part of this, because if you're not representing yourself, there's not going to be someone else who can do your job for you. And so as the research community is trying to work with you and your family, it's important to be counted and make sure that you're standing up for your child and putting their data into this. And I will emphasize that all of these data I'm talking about, everything in Simon's VIP, any data, any samples are freely available to all researchers around the world. So this is meant to be able to capitalize on your hard work that you do in terms of providing this. So I'm going to focus today mostly in terms of the way this channel affects the brain, starting with what we call developmental milestones, or when children first learn to do various different things. On the left, you're going to see the age at which they gave their first words or used their first words, and on the right, the age at which they first started walking. And you can see on the left side that what we call y-axis, that age, and every single dot is a different child. So you can see the age ranged in terms of when they first started either talking or walking. But one of the things that is also true is that not everyone has yet achieved either walking or talking. And so just to remind folks, all this is going to be available afterwards. But when you look at on the left or on the right with that text, there are individuals that you'll be able to read through and see have not yet started talking or walking. So as an example for the talking, there are 20 children so far who have not yet talked. And on the right, you'll see that there are 15 children so far who have not yet walked, who would have been old enough potentially to be able to talk or walk had they been quote unquote neurotypical. There are also some children, actually 5 of each, who were too young simply to be able to talk or walk. And so within this, you'll see that there's a range, and that's common with all the slides you're going to be seeing. There's a range and there are differences. And some of what researchers are trying to do is understand why there are differences and to a certain extent why the different mutations are accounting for some of those differences. In addition to that, we took information from you about the different behavioral issues or the different behavioral diagnoses that the children might have. The most common, I would say everyone had something in terms of some challenge. Certain individuals had more than one. So if you look at the number on the right, it doesn't add up to the total number of 52 children who were reported here because some people had more than one diagnosis or more than one label. The most common was either intellectual disability or developmental delay. That label depended largely in terms of how old the child was. And as Leah was saying earlier, some individuals have autism. A little less than half of the individuals have autism. But in another common condition, which oftentimes traveled with intellectual disability or developmental delay, were problems with language. And you could see that from some of the problems in terms of talking. Much less frequent were other conditions, but you can see listed here attention deficit, hyperactivity, anxiety, coordination disorders, other learning disabilities, but there were some other infrequent conditions. So I also want to tell you about some of the medical challenges. One of the things I want you to keep in mind, and the reason we put the ages of these individuals here, is that we're definitely skewed at this point to having individuals who are younger ages, and we have relatively few individuals who are adults. And so over time, I think we still have a lot to learn in terms of what some of the challenges are like as individuals age and get older. Again, starting with the things that are most common, brain issues or neurological issues were the most common. A large majority of individuals had problems with low muscle tone or hypotonia, also problems in terms of coordination, so problems with movement and coordinating movement such that they appeared clumsy. And the opposite of hypotonia or low muscle tone, some folks had high muscle tone or hypertonia. Those are the most common things. Again, these are not mutually exclusive. Folks could have more than one thing, and some of the other things that we saw that were less frequent were either smaller or larger head size. Some individuals were diagnosed with cerebral palsy, although I would actually call that a misdiagnosis. And then some of the things that can be seen in others, but headaches, for instance, and meningitis, which is not necessarily, I don't think, as common with this, but was diagnosed in one individual. The big thing, which many of you know, is that although it's not everyone with SCN2A, it's certainly an extremely common manifestation to have seizures or epilepsy. So again, out of the 52 individuals, in terms of having some type of seizure, the majority, or 40 individuals, had epilepsy. And I'm showing two columns. The column on the left is ever having that particular seizure type. And the column on the right are individuals who had what I would call medically refractory or high frequency or very frequent seizures. And so you can see on the right, it's not everyone, but there are several individuals who continue to have certain seizure types. Now, of those seizure types, febrile seizures, we as doctors oftentimes don't necessarily worry about that so much, but the other types that you're seeing listed here, the grand mal, petit mal, spasms, infantile spasms, complex, simple, all of those epilepsies or seizure types, I should say, are ones that we would try and medicate or try and treat for. Next slide. I list here, we've been collecting information from you about the different medications that are being used. And to me, it was important to put this because the condition was serious enough that it needed to have medication. And not surprisingly, you'll see that the most frequent type of medication that children are taking are medications for seizures. So anti-epileptics or sedatives or even cannabinoid oil or medical marijuana has been frequently used by families or individuals. Sleep issues were also not uncommon, and so several individuals have used melatonin for sleep. And then one of the other common findings are tummy problems, so gastrointestinal problems, either problems, as I'll get to, with reflux or constipation. And so those were the most common types of things. Again, a few individuals taking medications for behavioral issues, but the good thing is beyond those core issues, not a lot of other medical things. I know that's enough, but those are the core features, and I'll summarize that back at the end. One of the things that I know the families are most interested in, and we're also trying to gather this as best as we can, although it's not obvious, is what medications seem to be most effective or what medications seem not to be effective. The reason I say that it's not clear, at least to me, from what the families have reported back, is because we ask this in two ways. We've asked the families if they can either endorse that there is a medication that seems to be effective, and you can see that there are 4 different medications, not very large numbers in terms of the number of families reporting that they're very effective. And then we ask on the other side, are there any medications that seem not to be effective and that basically, you know, you've discontinued or you've stopped using because they weren't effective. And you can see that, in fact, some of the same medications that were reported to be effective for some parents were also stopped or reported to be ineffective by other families. And so I think we still have a lot to learn here. It's one of the things that obviously is most salient in terms of trying to get answers, but I don't think we have this figured out yet. Within this, and again, I'm going relatively quickly, just focus on the things that I think are probably most relevant, and you can see any of this online later. Do pay attention to some visual issues. Again, it wasn't necessarily the majority, but I will say that some of the children had some visual issues, some in the eyes and some really that are centered in the brain, but important if there is something in the eyes that can be corrected to go ahead and correct that. And again, as I was saying, there are some individuals who have what we call cortical visual blindness, and that's really a brain as opposed to an eye problem. And that brain problem isn't necessarily going to respond just to glasses, for instance, or something else in terms of surgery on the eyes or something else, but important to know and pay attention to with your doctors. Again, for the gastrointestinal issues, mostly reflux or heartburn and constipation. When we ask families in terms of diets, many of you have tried diets to be able to try and treat that. A variety of diets used either for the stomach issues or in some cases ketogenic diets to be able to help with the seizures. And some benefit, but I wouldn't say necessarily there's one sort of obvious diet to go to that's fixing for everyone, but some did report benefit with these. I'll just say very briefly, otherwise people reported a few other things, but none of them very serious or none necessarily very common. So I'd say the usual amount of infections, the usual amount of things like asthma, occasional pneumonias, relatively low, but some issues with, for instance, high sort of fast heart rate or high blood pressure, some relatively infrequent things in terms of a birth defect called hypospadias, easily treated, or a testicle that was rotated, some relatively minor kidney issues. And again, I don't mean to fly by, but these weren't very common, so I won't spend too much time on it. A few problems with gaining weight and being on the shorter side, and a few problems or a few common surgeries in general. Some of the growth problems or feeding problems treated with gastrostomy tubes for a period of time, or often ear tubes for the ear infections. One of the other issues just to pay attention to is when the children were especially growing rapidly, scoliosis. So do pay attention to that with your pediatrician. And then otherwise, relatively other common things. So in summary, not surprisingly, based on what we know about what this gene does, most of the issues are neurological or in the brain. That's where we're going to focus a lot more of our attention to. And otherwise, the way I describe it is really focus the attention there. The other issues, not so medically impactful. Just very briefly, again, to remember that all of this information, and for those of you who are kind enough to give blood samples, the cell lines of blood samples, all freely available to the research community. So let me stop there, and I think we've got a chance now for some questions.
All right, thank you, Wendy. So just a reminder to everyone joining the meeting, you can chat in any questions you might have for either Wendy or Leah, and we'll address them. Our first question that we have is whether or not we're tracking data on the kids that were walking and talking but then lost that skill.
So we are tracking that data. We have flagged that as what we call regression. So individuals who had a skill and then lost that skill. And as I think many of you know, that gets complicated because it's oftentimes associated with the epilepsy and the epilepsy not being under control. But the answer is yes, we are. And because we are doing this longitudinally in terms of checking in with the families once a year, we should be able to see how things are progressing over time. So for any individual, seeing multiple data points over time.
Okay, and we have another question. For problem gaining weight, do you find kids with diabetes or other metabolic issues?
So diabetes specifically has not been something that's generally been an issue. Again, remembering that individuals are young, but at least so far diabetes hasn't been an issue. The problems with gaining weight, I think, are a few different things. I think some of it is related to being able to eat, you know, sucking and swallowing and being able to coordinate all of that is part of it. This hasn't been something where everyone has consistently been, for instance, growth hormone deficient, or that people have needed to be on growth hormone to do this. In general, it's looked like the response has been if families have been able to get enough calories in or getting enough nutrition in, that people are growing appropriately in response to that. So I don't think it's necessarily primarily an endocrine problem, but more being able to get the appropriate nutrition in and figuring out ways to do that.
Okay, another question. You mentioned that in addition to having SCN2A, some children also have another diagnosis. Can you share what some of them are?
Either I may have misspoken or someone may have misunderstood, but we have at least tried to make this simple, if you will, in terms of the families that I've presented today really have, from a genetic point of view, SCN2A and only SCN2A. So we haven't taken individuals who've had multiple genetic diagnoses. What I was referring to in some cases is that some people from a neurological point of view, for instance, may have more than one neurological diagnosis. So they could have autism, they could have developmental delay, intellectual disability, attention deficit disorder, and they can have multiple diagnoses in terms of those either neurological diagnoses or behavioral diagnoses. But all of what I presented today is from a community that, again, from a genetic point of view, only has SCN2A.
Okay, thank you. And I know we were running a bit late, so I'll just do one more question before we move on to the next presenter. And this is a question just asking a little bit more about Simon's VIP and the research and asking about joining the research. So Wendy, could you talk a little bit more about just joining Simon's VIP and for those that may not?
Sure, very good. So with this, you can easily Google Simon's VIP or simonsvipconnect.org is the entire URL. As you do that, you can search for SCN2A. So we've got a page, if you look at what we study, or genes we study, you'll be able to see a page dedicated to SCN2A, and from there you can register to start joining the research study. You can register just to be part of the community, or you can register to start reporting and contributing data. If you do that, it's relatively easy. We try to make it relatively easy. You can do it from home, online, as well as then we have some of my genetic counseling team who can help you in terms of submitting your test reports that we review to make sure that you, in fact, do have the SCN2A and that we can get the nomenclature or get the exact mutation correct. And then we collect information from you at what I call baseline, so when you join the study, as well as on an annual basis. We try and make it relatively easy, and you don't have to do everything at once, so you can do it when it's convenient for you, at times it's convenient for you. But the majority of it is online to be able to do. And as I said, you can sort of stop and come back when you need to. But we do also have Ashley Wilson as a genetic counselor to be able to talk to you so that she can even talk through some of the issues or questions that you might have as she's collecting some of the medical information. Okay, thank you, Wendy. And unfortunately, that's all the time we have for questions right now. I know that some other folks did have questions, and we'll work on combining those and getting you all answers out after the meeting or if there's time later. But for now, I'm going to go ahead to our next presenter. So our third presenter is Dr. Stephan Sanders. He works at the Department of Psychiatry for the University of San Francisco. So go ahead, Dr. Sanders.
Thanks very much. I don't think I've got control over the slides, so I'll say next slide when I need them. So I'm a pediatrician who's then trained as a geneticist and bioinformatician, so I work with large amounts of data, and it was through doing that in autism that my interest in SCN2A started. I'd like to talk today about where we are from a basic science point of view and also starting to think about what a treatment might look like. Could I have the next slide, please? Great. So Wendy just gave a fantastic overview of the symptoms, and I must say I learned quite a bit from seeing that. This really is an emerging story where the data which families are submitting is making a difference in real time. It really is useful. When we look at the big picture of it, we notice three major patterns in the symptoms. There's a group of children who have seizures really starting in the very first few months. And those lead to severe developmental delay and many of the serious problems which Wendy was talking about there. And that's shown on the far left. There's a group in the middle who have seizures starting in the first few months of life, but then they tend to run in the family and they often resolve by 2 years of age. I'm going to guess that there's not many people on the call today who fit into that box because they resolve, and so that there's less reason to engage with the community like this. And then on the far right, we have children who present either with autism or with developmental delay or cognitive impairment, intellectual disability. And those children sometimes have seizures coming on later in life, for example, after 2 or 3 years. And those are the major three groups we see. And I think talking to patients, I think most children or most families we've met fit into one of those boxes. But there are some things we really can't fit into this. So, for example, Go on to the next slide, please. To understand why those three groups are there, we need to understand what SCN2A does. And so here I'd like to take you through a neuron in the brain and how that is impacted by this sodium channel called SCN2A. So a little bit of notation. Up on the top left, we have the name which we're all familiar with, SCN2A. That is the name of the gene which is found in the DNA. The thing which genes do is to make a protein, and the protein is called NAV1.2. Na is for sodium, V is it's a voltage-gated channel, and 1.2 is just the nomenclature because there's several different voltage-gated sodium channels in the brain. In this picture here, we're seeing a picture of a neuron. Specifically, it's an excitatory or pyramidal or glutamatergic neuron. They all, in this context, mean about the same thing. We see the nucleus, that's the big black bit. That's where your DNA is stored in the form of chromosomes, and that's where we have two copies of SCN2A. In the children who are affected, one of those copies has a mutation. The SCN2A gene makes RNA, which then goes out of the nucleus, and you get lots and lots of copies from RNA from a single copy of the DNA. That RNA goes into the soma there, and then it hits a ribosome where it is made into the protein called NAV1.2. And each copy of RNA makes multiple proteins. And so the whole thing amplifies, and that's why this single base pair can amplify to cause a problem at the level of an entire neuron. And of course, this is happening in all of the neurons, and that's why you get a problem at the level of the child. The sodium channel itself is mainly found in a place called the axon initial segment. So that's underneath the soma. So the big picture here is the neuron talks to other neurons through the axon, and that's the bit going downwards. So that might reach across the brain, and it might talk to another neuron, which then might do something to do with thinking or sociability or motor skills. Neurons also receive data, and that happens through the dendrites. And we're showing three processes coming out with dendrites on them. And it's on those dendrites that the synapses are found, and the synapses pass the data from the last cell to this cell, and then it receives it, and that makes it act. And so its axon will go down and connect with other neurons' dendrites. And the whole thing is basically like an electric circuit. So we find SCN2A at the start of the axon initial segment, and this is a very, very important place because it's here that action potentials are created. And an action potential is when a neuron fires and talks to another neuron. And in these neurons, which are, I think, the most common neuron in the brain, at least one of them, this sodium channel, SCN2A, is the sole creator of these action potentials in the first 2 years of life. And so it really, every single time a neuron fires, so this channel SCN2A is involved, and that's why it causes such profound problems. I'm showing here a little picture of the sodium channel. It's sitting in a membrane, that's the horizontal 2 lines, and it's shown in a closed state, and so normally it doesn't let sodium through. And there's lots of sodium outside the cell, there's not very much sodium inside. And then finally, I'm showing a little picture of the structure of it. A.J. Campbell, working at Harvard, recently worked out the structure of SCN2A, which is really important for drug discovery. So that's the big picture of how a neuron works. Let's go to the next slide. So here we're going back to a simple version. Now I will simplify the information and show you how the neuron works and what this channel does. So this is the neuron in its resting state. It's there, it's listening out for other neurons talking to it, but it's not doing anything particularly special at this moment. And the channel is closed. Next slide. Now, other neurons have fired, and it's detected that in its dendrites. So the synapse passes on neurotransmitters, and the dendrites receives it, and it starts to react. And the way it reacts is by the membrane potential, so it's voltage changing. And so the whole way which neurons work is through voltage. And so its voltage starts to rise. Let's go to the next slide. The voltage rises. SCN2A is a voltage-gated sodium channel. It detects that change in voltage, and it opens. And as it opens, sodium rushes into the cell. And as it rushes in, it's like putting a battery into a circuit. That flow changes the voltage. It makes it rocket up, and that's what an action potential is shown in red. Next slide. Milliseconds after it opens, something at the bottom swings over and shuts it. So the sodium rushes in, then it stops, then potassium rushes back, and the voltage goes back to normal. And that's why you get this peak, which is the action potential. Next slide. Which rushes down the axon, and then that stimulates the next cell through synapses and dendrites. And that's called forward propagation. But we also see back propagation, and that's where the action potential goes backwards towards the dendrites. This is less well understood, but the expectation is that this is how it trains the synapses and dendrites to work out how sensitive they should be, which is probably an important part of learning. Next slide. Then the whole thing resets and it's ready to go. And so that's the role, as far as we know, the sole role of SCN2A is to detect a change in voltage, open allowing sodium in, and kicking off this action potential. Next slide, please. So going back here, we have these three groups, the infantile epilepsies with early onset seizures, and autism developmental delay with often without seizures, but sometimes late onset seizures. Next slide. And we've, working as a group, we've discovered this, that we can understand this at the level of the channel. So on the far right, if you don't have enough channel, for example, you have a nonsense or frame shift mutation or a loss of function mutation, without enough channel, it's just not sensitive enough. And that seems to lead toward autism. And then on the far right, if the channel is too sensitive, if it opens at the drop of a hat, that means there's too many action potentials, which tends to lead towards seizures. Now, this is a model. It's not the complete answer. It's just a useful conceptual framework to think about it. It probably explains upwards of 80% of cases, but there are some which we're still trying to fit into this, particularly children with seizures between 3 and 12 months. Next slide, please. And so this means that if you have the gain of function, that leads to too many action potentials. They're hyperexcitable, which starts to sound like a seizure, and that's probably what begins the seizures. And then on the far right, we have in autism developmental delay, loss of function with too few action potentials, so they're not reactive enough, possibly causing a deficit in learning. Next slide, please. And so if we focus on gain of function infantile epilepsy for a second, we can see here that we've got the issues of forward propagation and back propagation. We believe that the main problem in these is an increase in excitability in the excitatory neurons, and this is probably the root cause of the seizures. If you've got too many action potentials, you can imagine the system reacts by having a seizure. And of course, the anti-epileptics act at that level. They try and reduce the excitability of the neurons, and we found that in these disorders with seizures before the age of 3 months, that anti-epileptics which block sodium channels are particularly effective. I should have given the reference there of the Wolf paper in 2017. Our hope is that future genetic or small molecule therapies would also have this effect. Now that's the initial issue. What we are less clear about is what happens next. So we think that this causes the seizures, but what do the seizures do to the brain? Does having these seizures and this change in excitability, does it change the way that neural circuits are made? This excitability is how the brain self-organizes and self-wires. And so there's a concern that that actually might lead towards a developmental delay. And then there's also a question of whether back propagation does anything. I think this is completely unexplored territory in the context of gain-of-function mutations. Is there a question here of it enforcing the circuit? So every time you have a seizure, it trains the circuit to say, I should do that again. This seems to be a route to getting more action potentials. And that actually those seizures themselves actually create a circuit which becomes more prone to seizures in the future. And that has important implications in terms of reversibility, because what we don't know is if we could magically make the neuron go back to normal, what would it have done to the circuit or to the training of the circuit? Next slide, please. Next slide, please. And then in autism, so here that we have the loss of function variants. Here we think that one of the main issues is reduced excitability before the first birthday. Now, unlike the increased excitability, this seems to go away around the age of 1 year. We actually understand why that is. It's because another channel comes and takes over. And so in the autism side of things, we have two questions. Firstly, does that reduced excitability, is that the major issue? Is it all during the fetal development in the first year of life? And does that lead to changes in neural circuits, essentially a wiring deficit, which leads to developmental delay? And that seemed to be in last year, I think we would have said that was our main answer to this question. There are two papers which were released this year, one of them by Kevin Bender in bioRxiv, another by the Middleton Group, and those are suggesting that back propagation plays a role. And so if you look after the first birthday, you find that the ability of these action potentials to train the circuit in its own cell actually becomes impaired, which might represent a deficit in learning, which could also lead to developmental delay. The question which we've not answered and are working on is which one of these two effects is the main route towards autism or developmental delay. And of course, there's also a question of it maybe being both. And the reason we are so focused on this is because at a simple level, changing wiring sounds quite difficult, whereas changing the function of the channel in the here and now about how it learns maybe sounds a little bit easier from a therapeutic point of view. And then I think we have another question is, why do children with these loss of function variants sometimes have seizures which come on late? The simple answer to that is we don't know. There's a paper out a couple of months ago which shows that it is the excitatory neurons leading to that, which is unusual for seizures, and also the mouse model which gives some insight into this. That's really useful because if the mouse has seizures, then it's much easier to test a mouse than it is to test medications on children, and so we might get some insight there. In the mouse, ethosuximide seemed particularly effective. Next slide, please. And so then we have the questions, the major questions to address. I'd sent a couple more slides, which I'm afraid have not made it into here. I'll try and talk them through because they really revolve around these questions and some additional questions Leah asked me about them. But I've shown what we're seeing there from the basic science point of view. The key questions we have now is what can be reversed. In 2007, in Rett syndrome, which is a severe form of developmental delay, there was a landmark paper where they used a clever mouse model where you could turn off the genetic deficit later in life. And they showed that turning it off in a mouse, which was the equivalent of a sort of 2 or 3 year old child, was able to stop many of the problems you saw in Rett, particularly mortality. And so that gave an idea that you can reverse something later in life. I think in the next year, we're going to see similar experiments being done in SCN2A, and I know that there are groups working on that, both for gain and loss of function. That gives us a really keen insight because it starts to talk about whether this is a wiring deficit or something which can be modified with the drug right now. I think there's a next question related to that, and that is how much does age affect reversibility? It seems logical that treating a 20-year-old is going to be much harder than treating a 1-year-old, which may be harder than treating in utero. We also then have some questions about how you might modify these questions. I'm afraid I've just been joined by a 2-year-old who's going to probably start screaming. Hello, Celeste. Bye-bye. Bye-bye. So in terms of the methods, there are three approaches which different groups are pursuing. One of them is looking at small molecules. These are things which could be taken orally. You might take them once or twice a day, and the hope is they would regulate the effect of the sodium channel, either increasing or decreasing how responsive it is. There's then several different genetic therapies, including antisense oligonucleotides, and then virally delivered methods to either upregulate or change the mutations. These are highly experimental. There was a landmark paper in 2017 which showed that those two approaches could be used in a disorder called spinal muscular atrophy, which is fatal at young ages. They were incredible papers. They have absolutely stimulated the field. I'm sure we were not alone in looking at those and thinking this could potentially change the whole of treatment of developmental delay. However, they are so cutting edge that there are some fundamental questions about the FDA about how we can approach these. The FDA is there to stop us from causing problems. They are vital in doing this. Cutting-edge therapies do not always work out the way you want to do them, and there are a couple of examples where scientists really, or clinicians did not do well with this. I think haemophilia rings a bell, where in the 1980s, huge numbers of individuals were given HIV through transfusion and unfortunately passed away. And then 14 years ago, there was an important case of gene therapy stimulating a very profound immune reaction. Those lessons have been learned, but we need to be careful about the lessons we need to learn in the future, and that is why the FDA regulates things so strongly. For small molecules, we would need to do a phase 1 human trial. That could potentially happen quite soon. Those cost on average $4 million and take at least a year. And then from a genetic therapy point of view, this is much more unexplored. You can't test those in healthy adults because they create semi-permanent changes. So we have to do work in monkeys, or what we call non-human primates. The cost of those is less known. We're working with the FDA to understand exactly what needs to be done to show them that these are safe. And I think the price point there is probably 5 to 10 million, if not more. And then there's an important question with the genetic therapies. Is that 5 to 10 million per mutation? Or is that for the concept of treating this? And that, again, is an unanswered question because it's cutting edge. And then the last issue we have is how would we measure success? In Rett syndrome, it was measured on the basis of survival. Now, that is an issue in some children with SCN2A, but it's not quite as severe as Rett syndrome and probably not enough that we could use as an outcome to show success. We need to gather data on phenotypes, so the information you submit to the registry. And this is going to be vital for us to work out whether these treatments work, because when we do this, we get one shot at telling the FDA what we think matters, again, to try and keep us all honest. If we get that wrong, then we might have a therapy which does work but doesn't look like it on paper. And so I think the key message here, as Wendy said, please, please, please give us your data. We need to know who's out there, we need to know the mutations there, and we need to know these relationships between the symptoms and the genetics, and that will have real implications in terms of developing these therapies. And I think that's my last slide.
Okay, thank you so much, Dr. Sanders. Our next presenter is Dr. Al George, who is the chair of the Department of Pharmacology at Northwestern University.
Thank you very much. It's a pleasure to participate in the virtual conference today. I'm going to tell you about an exciting new program that just initiated this last week when we were alerted that we were going to receive funding from the National Institutes of Health to fund a new research center without walls that we are now going to focus on the CHANNELOPATHY-associated epilepsies. This is a program that NIH has to fund collaborative research centers that don't exist within a single institution, hence the term Centers Without Walls. Last year, they announced a call for applications for groups to come together to study the functional consequences of human genetic variation and variants that are associated with the epilepsies. And the main focus of this was to really determine the function of genetic variants for which we didn't understand whether they were disease-causing or not, perhaps using that information to enrich or refine genetic classification of those variants. The grant announcement also encouraged groups to use these platforms to study the pharmacology of genetic variants, to investigate that at multiple levels, such as neuronal networks in culture or whole animals. And eventually, this is something that I think will improve the understanding of basic mechanisms, but also help translate that information to improvements in genetic diagnosis and maybe prognosis and hopefully therapy. So this is just a little bit of background that I wanted to share with you to give the big picture and why we decided to focus entirely on a small group of ion channels for our center. This is an image that I think is representative of the scale of the problem. There are over 100 genes that have been associated with monogenic forms of epilepsy, genetic forms of epilepsy. These images that you're seeing are, the boxes are scaled, and each box represents a different gene, but the size of the box is proportional to the number of known genetic variants in the literature. And this doesn't count those that haven't been published, that may be soon to be published or maybe never to be published. But what is striking is on the left side of the graph is all of the ion channel genes associated with epilepsy, and on the right are all the non-ion channel genes. Ion channels represent about 40% of known genes of all the known variants associated with epilepsy. SCN2A is a prominent member of that group, as well as two other sodium channels, SCN1A and SCN8A. And there's some notable potassium channel genes, KCNQ2 in the far left corner in yellow. Here is the actual numbers as of a few months ago when I investigated this using the human gene mutation database. There are over 1,500 mutations in SCN1A, more than almost 240 in SCN2A, and again, this is a number that's been growing dramatically over the last couple of years. And you can see the remaining genes there. KCNQ1, which will be a focus early on of our center, is also a large notable gene. And overall, the growth of epilepsy-associated variants in just the voltage-gated sodium channels and voltage-gated potassium channels is just continually going up and has not reached a saturation point yet. So for these and many other reasons, and just to illustrate what is happening in the genetic diagnostic space, this paper, which came out in May of this year by the group at GeneDx, reported that if they looked at their a series of 8,000 cases for which they were applying genetic testing for, they identified among half of those cases, those that were positive for any mutations, half of those were in these 4 ion channel genes, 3 of which were sodium channels, and that includes SCN2A. And KCNQ1 was the lone potassium channel. So these, again, were data that suggested that a large fraction of epilepsy-associated variants were in genes encoding ion channels. Most of those were in sodium or potassium channels, and the opportunities to understand the functional consequences of those, as well as the pharmacological opportunities to improve treatment, were vast, and we and our colleagues were poised to attack this. And so this is the focus of our project. And so the premise of our study was, as I just mentioned, there are a small number of sodium and potassium channel genes that have the highest cumulative burden of genetic variants among the epilepsies, that these genes are targets for drugs that we have on the market now and others that are in development that may be therapeutic. And there's a lot of established approaches to do the functional consequence of those. And so those are sort of the main things that we set out to convince reviewers that we should be funded. And then just sort of the view from 5,000 feet, the objectives and the goals of our center are primarily to implement high throughput methodology to determine the functional consequence of as many genetic variants in sodium channels and potassium channels as we possibly can in 5 years. And we painted a lofty goal. We said, given that there are well over 2,000 variants known today, and I have no idea what that number will be in 5 years, that we should set our sights at doing something transformative. And so we've proposed to try to get to over at least up to 1,000 variants studied in 5 years. Now, that's not going to be entirely funded by this grant. This grant is big, but it's not that big. But we think through enhancements in our efficiency at doing the work, plus some additional funding from other agencies, foundations, philanthropy, we set our goals on 1,000 variants in 5 years. In addition, and so those high throughput methods are done in the most simplest system possible, a cultured cell in the laboratory. It's not a brain cell, it's not a neuronal cell, it's just a simple cell that doesn't have any of its own ion channels. And we, through the magic of molecular biology, we can introduce the channel that we want to study in the form we want to study it, as a mutation, as the normal version, and we can apply some very high throughput electrophysiological studies to actually crank out data on dozens of mutations at a time. Now, that's the most primary approach that we have. The next elements in the study were to actually take that information and compare it to what we would learn in a more complex cell system or in an animal. And so we have the high throughput part of the study, and we also have the high content part of the study, where we're going to be using two approaches. One approach using modern technology to take a stem cell and differentiate it into a neuron that we can study in the laboratory. And then beyond that are plans to make genetically engineered mice that have specific mutations that we can also study at a whole organism or whole brain level. And eventually we want to continually put this information out. We, of course, will be publishing papers, but even before we get to the point of publishing, we want to figure out a way that we can disseminate this information on from all levels of our research, particularly the high throughput studies that will inform us as to the basic functional consequence of a genetic variant. And our goal is to eventually try to get that information as soon as we can into a publicly accessible database, such as the ClinVar database, even before papers are written. And that's what we're really committed to doing, so the information is widely available and hopefully will improve the diagnostic accuracy and value of genetic testing going forward. So here are some of the key questions that our center is trying to identify. I'm not going to read all of those, but I think one of the drivers for much of this work is trying to understand why sometimes the same mutation or similar mutations in the same gene cause very different epilepsy syndromes, or sometimes very different severity of syndromes, or why mutations in the same gene may result in really completely different clinical manifestations. We want to make sure that when we study a genetic variant in one system, that we can predict or get good correlation with what we observe in another system, say in a stem cell-derived neuron or a transgenic mouse of some sort. And then we want to investigate the pharmacology of the dysfunctional channel variants in trying to understand, is there a best drug or combination of drugs for each variant. And right now we don't know that. We have some suspicions that there may be variant-specific pharmacology that would be very valuable to delineate. So our main aims are listed here. Again, I think I've sort of covered that. We're mainly interested in conducting this very large scale, sort of unprecedented scale of functional evaluation of ion channel variants with a focus on sodium and potassium channels. We want to look at whether that pattern of ion channel dysfunction correlates well across more complex models for investigating these mutations, and then to disseminate the information. Here are the participants in our Center Without Walls. There are 5 academic centers listed here. Northwestern is the center of the center and the primary home for the grant, and we'll be directing the traffic. We have an important and strategic collaboration with the Broad Institute, who has some similar high throughput technologies that we're going to couple with our effort. Baylor College of Medicine, which is where Ed Cooper's laboratory is located, and he's been a real driver in KCNQ2 studies, which is one of the important epileptic encephalopathies. University of Connecticut with an interest in KCNQ2 as well. And Indiana University, where a colleague of ours who is an expert in sodium channels, Ted Cummins, is located. We also have 3 research hospitals involved. There's Children's Hospital Philadelphia, Lurie Children's Hospital Chicago, which is our immediate neighbor, and the Cleveland Clinic, where Dennis Lal has just moved, and I thought I saw Dennis's name on the call. Hi, Dennis. And then in addition to our academic partners and our research hospital partners, we have 2 industry partners, including GeneDx, which is a very important genetic diagnostic company, and Q-State Biosciences, which has some additional novel high-throughput technologies that we hope to exploit. And let's see. So the way our center is going to work is we actually have a group that's going to really evaluate and prioritize which genetic variants should we study first, and maintain that list actively to feed the research projects. And so we have a very important group of people, including Ed Cooper at Baylor, Ingo Helbig at Children's Hospital Philadelphia, and others who are going to prioritize the variants we should study based on whether they are recurrent variants, how they've been classified previously, and whether there's unique features about those, whether those are clinical features or in response to drug therapy. But they're the experts that we're going to depend on to actually give us the list annually of which variants we should study and in which genes. Then those variants are going to be studied functionally, and when the opportunity is available, we'll study those same variants in advanced cellular models such as stem cell-derived neurons, as well as mouse models, which will be engineered as part of the study. The list of genes on the left, which includes SCN2A, these are not in any particular order of priority. This is just the list of potassium channel genes and sodium channel genes. We do have a leaning in the first year of our project to do KCNQ2, and the reason for that is simply that this center of ours really was driven in part by a prior effort by Ed Cooper at Baylor to get a center funded around entirely around KCNQ2, and we've sort of incorporated that team effort into our effort, and therefore we're giving a little bit of a head start to KCNQ2 for a variety of reasons. But SCN2A is on our list, and we hope that we will be actively studying SCN2A in the near term as well as long term, and there again we rely on our variant prioritization team to give us guidance about that. I think I'm almost done. I just want to point out that there are 3 scientific projects in the center, and those are listed here. Project 1 is really the major high throughput functional evaluation project, and that's going to be led by myself and Jen Pan at the Broad Institute. Project 2 is focused on developing human neuron models of epilepsy caused by ion channel mutations, and this is going to be led by my Northwestern colleague Evangelos Kiskinis and Owen McManus, who is a scientist at Q-State Biosciences. So this is an unusual partnership of an academic and an industry group. And then lastly, Project 3 is going to be targeting developing novel mouse models of epilepsy caused by channel mutations. This is led by my colleague at Northwestern, Jennifer Kearney, and Ted Cummins at Indiana University. We've already begun to make Jennifer Kearney has begun to make unique models of SCN2A mutations in mice, and that's something I think will be a high priority of the project. Then supporting the scientific projects, we have 2 scientific cores, which simply means shared resources that are supporting the activities in the scientific projects. I mentioned the variant prioritization core. This is led by Ed Cooper and Ingo Helbig. Their job is to figure out what variants we should study and why. And then on the back end, when we have functional or pharmacological information that we want to make public, they're going to be the ones important to curating that information and getting it deposited in ClinVar or other appropriate databases so that everyone can see it even well before we publish the work. The second core is really the workhorse of making all these genetic variants for functional studies, and that's going to be led by Carlos Vanoy here at Northwestern and Jen Pan at the Broad Institute. And then an administrative core, which really is there to kind of glue everybody together and kind of keep the pace. Myself, Ann Berg, and John Millichap are going to be involved in that. Ann and John are both at Lurie Children's Hospital in Chicago, and their main job is going to be to help our whole center engage the community of families like yourselves on this call, as well as primary care providers and geneticists and genetic testing companies who are interested in this information, and keep them informed as to what we're doing at all steps of the process. And so it's a very integrated group of projects and cores. This is not such an important slide to hold on to, but I also want to say that each of these individual elements of the project is really a group effort, and we've really assembled a team of people that really want to work together, in some cases have a history of working together. It's going to be a highly collaborative project. Multiple people are involved in each project in core, often across institutions, and so this is truly a center without walls. And lastly, and a lot of thanks to both the SCN2A Foundation and the other family foundations that helped write important support letters for our application. And we're not going to forget you. We're going to continue to remain involved in what you're trying to do and your missions. And we also want to have multiple webinars like we're doing now to sort of provide information about what we're finding and how the data can be shared and get your input. So that's all I had to say. Thank you.
Okay, thank you so much, Dr. George. We're going to have some time now for some Q&A, so remember to use that chat feature if you do have questions. The first question is for Dr. Sanders, which is just, are you able to share the table that you cited in the TINS publication?
Yes, absolutely. We made it open access, so it should be possible to download it straight from that website. I can share it with anyone who wants it. Absolutely.
Perfect. Okay. And the next question is, do the numbers of SCN2A mutations reflect, and I think this is for Dr. George, does the number of SCN2A mutations reflect how many variants or the actual cases?
The number of variants are the number that I showed in that table reflects the number of individual variants, not cases. Number of cases is likely to be much higher.
Okay, thank you. And then another question for Dr. George, how many models of each gene will be created, and will they be shared with other researchers?
Yeah, so I think the first project is going to be making cellular, we'll be doing experiments in cells, and there's not going to be any real durable and shareable model per se, although we will make all of the plasmid DNA constructs freely available to people. In some cases, we will make stable cell lines, which could be used for drug screening or other experiments, and those will be made available as well. I can't give you the exact number, but the number of plasmids will be probably close to 1,000. The number of stable cell lines will probably be a couple dozen. In terms of the induced pluripotent stem cell-derived neuron models, that number is probably going to be in the range of 10 to 12, and those also, we intend to share those. Now, a caveat is that many of those cell lines are first created by taking a sample, usually of blood, from an individual who has the mutation, and that's done under informed consent here at Lurie Children's Hospital. And so I believe that it's been set up so that we can share the cell lines, but we would probably have some restrictions in terms of who would be able to receive them. So I think academic labs would be able to receive them as long as they have appropriate approvals to work with them. Commercial entities is more complicated. The third type of model we're making are mouse models, and those will be all shared freely. There are repositories that we will target to deposit the mice, including the Jackson Laboratories, for example, which takes in mouse models from academic investigators and makes them available with a nominal fee for the cost of providing the animals. So, and again, the number of mouse models is probably going to be in the order of about 10 to 12 different genetic variants, different mouse models.
Okay, and then I have a question that came, I think, toward the beginning of Dr. Sanders' presentation, that what kind of risks can come with being able to reverse the gene, and what successes have you all seen with other genetic therapy?
In terms of the risks, for small molecule, it depends where on off-target effects. Probably the biggest one for small molecules is there's a very similar channel in the heart called NAV1.5, and the concern would be that something which targets SCN2A also targets the heart version, and that could lead to funny rhythms in the heart. And so that's where it needs extensive testing in mice, in a dish, and then also in healthy adult humans. There's then also the possibility it causes something which no one has thought of, and that's why it needs these safety trials. With the genetic therapies, probably the first one is immune response. So a lot of these are delivered by viruses. The new adenoviruses provoke much, much less of an immune response, and I think there's much more awareness that it's a potential concern. There are precedents of some of those being given in humans. The number of precedents are small because this is cutting-edge work. There's also then the theoretical concern that if you did truly reverse it, could that cause a problem itself? We worry a little bit about overshooting. So if someone's got seizures, if we dampen it down too much, do we cause developmental delay in autism? And if someone's got autism or developmental delay, if we overcorrect, does that cause seizures? We, as a community, just need to be very, very careful about testing it enough to try and evaluate whether those safety concerns are evident. Did that answer? Was there a second? Sorry, was there a second part to that? I've already forgotten it.
I think that was it.
And that really is why it takes time.
Okay, and then we'll have time for one more question. This is, how can families in our group help with the research for CWOW?
I guess that's a question for me. So I think that we, again, through our community engagement plans, we hope to get some feedback from families that have a specific interest in each of the genes we're going to study. Some of that feedback could tell us about important unusual features of specific mutations that we really want to dive into in more depth. I think that's something we've imagined as being an important contributor. Separate from the grant, again, at Lurie Children's Hospital, we have a program to collect blood samples for making induced pluripotent stem cells from patients who have known genetic mutations in the ion channel genes we're interested in. That's a program that's been active now for 2 years, and we're closing in on well over 40 independent iPS cell lines that we've already made. It's something that we still want to build, and so families who want to participate in that study can email me, and I can put you in touch with the genetic counselor at Lurie Children's Hospital, who is the kind of study coordinator for that.
Okay, thank you so much. We're going to move along to our next set of presenters. So first up is Dr. Keith Coffman. He's the director of the Movement Disorders Program at Children's Mercy Hospital, the director of the Tourette Syndrome Center of Excellence, associate professor of pediatrics at the University of Missouri, Kansas City School of Medicine. So Keith, go ahead.
So it looks like I don't have control of the slides. So I, like Dr. Sandler, shall have to have folks advance for me. So if you go to the next slide, please. So we're going to switch gears and talk about the human condition and how folks with SCN2A are affected by this from a movement standpoint. I'm going to focus exclusively on the folks with the gain-of-function mutations. First, we're going to talk about the basal ganglia. So the basal ganglia are the internal brakes of the brain. They help to generate and inhibit movement. And when they malfunction, extra involuntary movements can emerge. Next slide, please. So as you heard from Dr. Sanders earlier, with a gain-of-function mutation in the SCN2A gene, neuronal excitability is increased, hence the development of epilepsy. The basal ganglia get the bulk of their input from the cortex of their brain. So therefore, when the cortex, the pyramidal neurons, as you heard him describe, when they are pathologically activated, the neural activity of the basal ganglia is disrupted as they receive their input from the cortex. Next slide. So here is a wiring diagram of the cortex of the brain down to the basal ganglia, to the thalamus, and then back to the motor areas. And this comes from Blumenfeld's neuroanatomy textbook. The blue structures that you see in the center are the basal ganglia, and you can see the neurotransmitters abbreviated in this slide. So GLU, at the top is glutamate, which excites the brain, then the basal ganglia. And you see GABA, and many anticonvulsants work on GABA receptors. And then you see other abbreviations, DA, which is dopamine, and ACH, which are acetylcholine. And this is the chemistry of the basal ganglia that help control movement. Next slide. So when we think about the topography of the basal ganglia, so the basal ganglia don't just inhibit movement. They actually have multiple parallel circuits that subtend motor function, sensory function, cognitive function, and limbic function, which is emotional control. And you can see the red box behind the center group of squares. This is the location where I, as a movement disorders doctor, mostly modulate people's neurology in order to get movement under control. Next slide. So when we think about a hyperkinetic movement disorder, this is a condition where there's an inability to control voluntary movement due to the presence of extra or unwanted movement. You heard earlier folks talk about hypertonicity. Hypertonicity, also known as spasticity, is only noted when the examiner moves the patient, not when the patient is moving. Whereas dystonia, which we'll focus mostly on, is a movement disorder, not an abnormality of muscle tone. Next slide. So dystonia is, on the planet, the third most common movement disorder. And it's characterized by body stiffening, contortion, and sometimes an apparent arrest of motor activity. And multiple regions of the basal ganglia are linked to dystonia. Dystonia can be just one body part, which is called focal. It can be multi-segmental, or it can be the entire body, which we call as generalized. And for those of you who know epilepsy, you heard partial, you heard generalized. Dystonia, like epilepsy, can be variable from patient to patient and even within the same patient. And dystonia, as you will see, can occur in patients without other neurologic disorders. Next slide. So dystonia neurologically is characterized by the simultaneous contraction of agonist and antagonist muscles. So what does that mean? That means muscles that flex a joint and extend a joint are locked at the same time. And they're locked sufficient to induce a posture. So patients with dystonia, and you'll see from the videos later, often have very unusual postures that look uncomfortable, as dystonia is very uncomfortable. When I'm seeing patients in the office, what I describe to them, dystonia, many people haven't heard of it, but when I say, have you ever had a Charlie horse? Yes, they have. That's actually a dystonic movement. because of the dystonia, many patients who have dystonia can have constipation and reflux. And you heard earlier that many patients with SCN2A have issues with GI motility. And is that because of the epilepsy? Is that because of their hypotonia? Or is it the fact that some of them may have under-recognized or unrecognized dystonia? Next slide. So we're going to show you videos of two different children who have SCN2A and have dystonia. Next slide. So if we could play the first video, please. You see this little girl. She is awake. She's alert. She has these unusual movements of her arms that look somewhat tremulous, but you see the movements are irregular. And they continue to shake, and she's shaking involuntarily. You can see that her mouth has this open type posture to it. So this is dystonia that's looking like tremor. We sometimes refer to it as dystonic tremor, but really it's dystonia that's more brief in terms of contraction. Next slide. Next video. So this young man, as you see, has a very significant abnormal posturing. He's got arching of his back called epispatonis, and you can see how uncomfortable he is. His face is red. He's got that same open mouth appearance that the young woman did, and this goes on for quite some time. There's another video of him, if we could show that, please. Next slide or next video, please. Oh, okay, it looks like we're having some difficulties getting the videos to play. So as you can see, dystonia is a painful thing to experience, both as a patient and as a parent. When you think about dystonia, what dystonia can do to the body if it's untreated, and you see I love alliteration, so it can deform body parts. So with those prolonged contractions over time, you can actually get joint remodeling and end up needing orthopedic surgery because of dystonia. You can have dehydration. So dystonia is a tremendously active process metabolically.
Dr. Coffman, are you still on the line? I apologize, folks. We may have lost audio here. Just bear with us for 1 second.
Hello, I'm back. Sorry, my phone cut out. Could we go back one slide? So dehydration can be a severe consequence of dystonia, and I have many patients who have been hospitalized and some in the intensive care unit with dystonia. And then delayed growth and development, because dystonia is a tremendous calorie burner. In essence, patients who are undergoing dystonic movements are doing physical activity equivalent to high-degree aerobics all the time. Next slide. When we look at the way we treat dystonia, it is in many ways similar to how epilepsy is treated, with benzodiazepines, and on the bottom you see anticonvulsants, some of which have been talked about today. And then in the middle of the slide there are other medications, baclofen, trihexyphenidyl, and carbidopa-levodopa, which work more on the basal ganglia to control the movement as opposed to the cortical level where the other medications work. Next slide. We're now going to talk about dystonia with dysautonomia, and this is a grossly under-recognized phenomenon in the pediatric community. It's also been known in the past as paroxysmal autonomic instability with dystonia, and this disorder is seen in patients with severe encephalopathic brain disease. These events are sometimes called thalamic storms, but in actuality, they're hypothalamic in origin. Next slide. So what happens with these events is you get the sudden onset of severe generalized dystonic posturing, and accompanied by hyperpyrexia, so fever, tachycardia, hypertension, tachypnea, and diaphoresis, which is sweating. Obviously, this is often mistaken for seizure, and these episodes can last minutes to multiple hours. The difference between them and epileptic seizures is often the patients who have them will be irritable and crying for obvious reasons during these. The second thing that sorts them out from seizure is that they do not respond to anticonvulsants and benzodiazepines. And lastly, during EEG recording, the EEG does not reveal any epileptic discharges during the movements. Next slide. So if we could show the first video there. So you see that's the same young woman from earlier, and you hear how uncomfortable she is, and you see her abnormal posturing. You see the deviation of the toes on her left foot pointing down, her flexed position of her arms, her twisting at the torso, and you can see how red she is compared to the first episode where she was just sitting there. Could we show the second video, please? So this, as you see, is the same young man from earlier. And what I want you to pay attention to is the redness of his face. So this redness, this severe reddening of his face, along with the dystonic posturing, this is an extraordinarily uncomfortable situation and can go unrecognized as just irritability or behavioral, or sometimes is he ill because he's red and he has a fever, but in fact this is an autonomic overflow resulting from problems with overexcitability in the brain, causing problems in the hypothalamus, leading to this phenomenon. Next slide. So abortive therapy for these events is absolutely ineffective, and prophylaxing is the key. There's little data, however, about what's effective in prophylaxing for this phenomenon, regardless of etiology. The medication that I've come to use over the past decade plus is clonidine, which decreases the release of norepinephrine in the brain. And there are other options if those fail, but there, as I said, very little evidence based in what to do to treat this. Next slide. And that's it for me.
Okay, thank you so much, Dr. Coffman. Our next presenter is Dr. Heather Olson. She's an assistant professor at the Department of Neurology for Boston Children's Hospital and instructor of neurology at Harvard Medical School.
Hi, thank you. This is Heather Olson. So I was asked to talk about the clinical spectrum and comorbidities associated with SCN2A related disease. You've heard some pieces of this along the way, and I'll try to fill in some additional pieces from my perspective. I'm in child neurologist with specialists in epilepsy and epilepsy genetics. Let's see if I have control of my slides. Can you all hear me okay?
Yes.
I'll assume yes. Okay, so I have some grant support, but this is actually a broad overview and not discussion of my own research. So we'll talk about and focus a lot on the epilepsy syndromes that can be associated with SCN2A pathogenic variants, a little bit of movement disorders, but you heard a lot of that just now from Dr. Coffman, and other comorbidities. So I wanted to talk first about a term that's been fairly recently proposed called developmental and epileptic encephalopathy. Epileptic encephalopathy is an older term. This is a term that's been more recently proposed to describe a group of infantile and child onset epilepsies characterized by developmental slowing or regression in the context of recurrent seizures and frequent interictal epileptiform discharges on a background of developmental delay. So sort of getting at the idea that there's a mixture of the seizures and a developmental picture being symptoms of an underlying encephalopathy or brain dysfunction, in this case related to a genetic disease, which is frequently the case. So examples include Dravet syndrome, which is one of the first identified genetic epilepsies associated with abnormalities in a cousin gene, SCN2A, as well as Ohtahara syndrome, a very early postnatal onset epilepsy, and West syndrome. I'll describe those in a little bit more detail here as well. So in patients with pathogenic SCN2A variants, there's a wide spectrum of epilepsy syndromes. And epilepsy syndromes, the electroclinical syndromes are described, or a description, each one of a group of seizure types, EEG patterns, and clinical features lumped together. Often age of onset comes into play, and developmental features as well. So with SCN2A, this is a list of the various syndromes that have been associated, and they can change over time as well, and I'll show you that. So benign infantile seizures was talked about a little bit earlier, and I'll go over each of these in a little bit more detail as well, but that's the most benign early onset type. Ohtahara syndrome, a more severe neonatal onset epileptic encephalopathy, a non-specific neonatal epileptic encephalopathy, so ones that don't have a name but are still in that category. Something called epilepsy of infancy with migrating focal seizures, West syndrome, Lennox-Gastaut syndrome, and children who have autism spectrum disorder and not an early epileptic encephalopathy, but have later onset epilepsy. So that's not quite an electroclinical syndrome, the latter one, and tends to fall in more of the loss of function group of patients. So these are, as I mentioned, age dependent. So it isn't necessarily one gene, one syndrome. So SCN2A is not unique in causing these electroclinical syndromes. And they often have to do with the age and developmental stage of the brain. So Ohtahara syndrome is often one of the early and severe epileptic encephalopathies in the first couple of months of life. In the mid-first year, you can get West syndrome, and later in early childhood, Lennox-Gastaut syndrome. And you can evolve from one to the other. For example, 75% of patients with Ohtahara evolve to West syndrome, whereas you can also get these syndromes without having a prior epileptic encephalopathy. So West syndrome can be the first start of your seizures, for example. So this is another figure that you've already seen today in Dr. Sanders' talk, just showing the relationship of some of these syndromes with the changes in the sodium channel and the dysfunction that you see related to the genetic variant. So in the group that have loss of function of the SCN2A protein, it's more often a picture of autism spectrum disorder or intellectual disability with or without seizures that often have onset in childhood or after the first year of life. Whereas on the other end, in the loss of function group to the left of your screen, there's a spectrum from the benign infantile seizures to the more severe epileptic encephalopathies, but most with very early onset, even just in the first life or two postnatally. So Ohtahara syndrome is one of those neonatal or just in the first month or two after birth epileptic encephalopathies with a specific EEG pattern called burst depression that I'm going to show you a picture of, not for anybody to feel like they need to learn EEGs, but just to put a picture in your mind on the next slide. And associated with predominant seizure type being something called tonic spasms, which is stiffening and clenching of the body for just a second or two at a time and then relaxing, and those often happen in clusters. Most of these patients have onset in their first month of life. This is a picture of that burst depression that I mentioned. If your child has an EEG, those little electrodes or buttons that they put on the head, each line here of squiggly lines is one electrode compared to another. And this is just to put a visual in your head of burst depression, which is periods with very sharp, high amplitude activity, looks kind of ugly, followed by periods that are very quiet, and that would be unchanging throughout the EEG, wake and sleep. Versus this is an EEG image of a child with a milder but still epileptic encephalopathy pattern with onset in that first month after life. The lines look less high amplitude and spiky. They're a little bit calmer, wiggly lines. And that's closer to what we expect for normal, still with a few sharp features here and there. Epilepsy of infancy with migrating focal seizures typically has onset by 6 months of age, somewhere in the first half of life, and that's a different pattern, usually not right away after birth. Patients presenting with focal seizures, often with a motor component of some twitching or stiffening and or some autonomic features, change in heart rate, change in breathing, sometimes with respiratory compromise. And in EEG, they have a very characteristic pattern where they actually migrate from one portion of the brain to another portion of the brain. So they migrate around, which is a pattern we don't typically see in other types of epilepsy. And that is typically associated with a challenging developmental outcome. West syndrome is typically onset about the mid-first year of life, somewhere around there, with a seizure pattern, infantile spasm, so similar to Ohtahara syndrome, but with later onset, and a different classic EEG pattern, something called hypsarrhythmia. Not going to show you that one. That's a little bit more complex, and in the setting of developmental regression as well, so it's a triad of symptoms. Lennox-Gastaut syndrome is a syndrome that often presents more in early childhood, or you can evolve to this pattern from an earlier epileptic encephalopathy, like that first picture I showed you. There are typically multiple seizure types, including drop attacks that can be either tonic with stiffening or atonic with loss of muscle tone, and is typically refractory to medication and associated with intellectual disability, as well as, again, some characteristic EEG patterns, something called slow spike wave and fast activity. And then there's the different spectrum of kids that tend to have loss of function mutations who present with autism spectrum disorder or intellectual disability and may or may not have later onset epilepsy. Those patients are a little bit underrepresented in some of our cohorts just because getting genetic testing in that cohort of patients has been more challenging even than in the epilepsy subgroup. And the other thing I wanted to mention was just that autism spectrum and ID certainly can be features of the children with the earlier onset epileptic encephalopathies as well. It's just that their epilepsy patterns differ. So movement disorders, you just heard a discussion of dystonia and actually a bit about the autonomic dysfunction that can go about with that as well. There are some other movement disorders that are described and can be present in patients with those epilepsy phenotypes or separately from epilepsy. So chorioathetosis, and I apologize, I don't have videos at some point with a longer talk. Videos would be great, is kind of a rising type movement of the extremities. Episodic ataxia is one that is more and more described in the spectrum of disorders, typically with onset after the first year of life, but can be in patients, again, with the epilepsies or without the epilepsies. And you can have the ataxia as a sort of a sudden change where the kids will become uncoordinated, their gait may become more wide-based, for example, difficulty reaching their targets when they're reaching for things, and that can be accompanied by some other features like slurred speech, nausea and vomiting, or pain, like head and back pain. myoclonus can either be epileptic or non-epileptic, but you can see non-epileptic myoclonus in some of these kids, which is if you think of the phenomenon of jerking awake as you're falling asleep, that's a benign form of myoclonus that many of us have, but you can get myoclonus throughout the day as well, and that can be a feature of these disorders. And then stereotypies are repetitive movements. They can be hand wringing, they can be flapping, they can be a variety of different, usually hand-type movements that kids do repetitively. Frequently see them with autism, but can see them separate from that as well. So thinking about some of the other comorbidities, hypotonia or low muscle tone is common. And some patients will have increased tone over time as well, whether that's the dystonia that we talked about already, or spasticity, which are just a description of different kinds of high tone. Sleep dysregulation is a big challenge in this population of kids. Both it can be falling asleep, it can be maintaining. Something to definitely think about and work on when it's a challenge. Swallowing dysfunction, especially in the kids with the lower muscle tone or the more severe epileptic encephalopathies, can be an issue, so trouble swallowing. Some kids will have risk of aspiration, which is getting the food or the liquid and down the wrong tube into the lungs. On the severe end, that can cause a pneumonia, but it can also be very subtle or silent. And kids may require thickening of liquids or gene tubes at times. And then GI symptoms, constipation and reflux are pretty common, which is a challenge to other genetic. Visual impairment is recognized, especially if it's on the milder end of the spectrum. But patients can have usually not a problem with the eyes or the structure of the eyes themselves, but the interpretation of the vision or the processing of vision in the brain, called cortical visual impairment. That's something that is amenable to therapy. And so it's something we pay close attention to. And then autonomic dysfunction. I'll have another slide on here, and you heard about autonomic dysfunction in the context of dystonia, and that is one of the situations where it definitely comes up. There has been concern for increased risk for arrhythmias, although there aren't great large studies to establish whether or not that's a problem unique to this disorder or not. And so that's something for future work. Temperature dysregulation is something we frequently hear reported, and episodic hyperventilation or fast breathing, and or apneas where you actually have longer pauses in your breathing. And then autonomic storming is something that typically happens in the setting of dystonic events, which is what Dr. Coffman was describing. I mentioned this briefly earlier, but just to emphasize, a lot of the literature and the experience that we have out there is related to children with the early seizure type and the gain of function variants or presumed gain of function variants in this SCN2A gene, in part because the autism cohort and the intellectual disability cohort are less often diagnosed, but they're definitely out there. And at our center, it's also a little bit skewed because I'm in the epilepsy genetics group, but we have discussed with our autism program, and there just are fewer diagnosed patients there as well. So a little bit of skewed experience. These are some of the faces of SCN2A, which is actually from your website. I will work on getting my own pictures up there. I just need the consent from the families. We have tons of cute kids, but all of these kids make it worth it, and they're why we keep trying to do better for them. So thank you very much. I have my email here, and this is a picture of my own cutie little daughter who is playing with her dad to let me take time to do this.
That was great. Thank you so much. We're now going to move into another question and answer period, and we're going to try to catch up on some of the questions from previous presenters as well. Dr. Sanders had to drop off of the call, so any questions for him I've been compiling, and I will follow up with him after the call. But for now, I'll just get going on some of these questions. So one came in for Dr. Coffman, which is that if you suspect your child has dystonia or dysautonomia, how should you go about getting that diagnosed?
So what I would say is to, as you saw, the videos that I provided were actually videos of patients taken by their family members. So because this phenomenology requires really the person to see it, get videos of the things you have in question, share them with your healthcare providers who are taking care of your child, and ask them to, assess the movements that you're seeing, and maybe they have, like we do at our institution, both movement disorder doctors and epilepsy doctors to be able to look at those videos and get a right diagnosis.
I apologize. Do people who have dystonia have them all the time with varying degrees, or can some only have dystonia during seizures?
So dystonia is variable. Some patients have dystonia throughout their entire waking day. Some patients only have it with certain attempts at movement. It's quite variable, which is why capturing it and really demonstrating what you're seeing in your child is the most important to be able to figure out, is it a normal child movement or is it abnormal and needs to be treated?
Okay. And then the next question, can dopamine help with movement disorders like tyrosine or a Parkinson drug?
So yes, you saw on my slides carbidopa, levodopa mentioned. That is the precursor to dopamine. It's commercially known as Sinemet, and it is one of the medications that I use the most to treat patients who have dystonia, whether it be from SCN2A or any of the other pediatric onset movement disorders.
Okay. In the slides, it's mentioned that EEG cannot record signal. Is this the reason it's called dystonia rather than a seizure?
Perfect question. No, the neurology of dystonia is best characterized by abnormal activity in the basal ganglia. So unlike epileptic seizures, where, as you saw in the EEG demonstrated, you see abnormal signals being generated on the surface of the brain. In dystonia, the abnormal activity is within the basal ganglia, which does not have the ability to generate epileptic seizures, but only abnormal movements.
Okay. The next question is, can you please explain other symptoms of autonomic dysfunction and when and how to treat these?
So I'll take a stab at it first. So autonomic dysfunction, as you saw from both my slides and Dr. Olson's slides, problems with temperature regulation, problems with sleep-wake cycles, problems with gut motility, meaning constipation or sometimes diarrhea, sweating, high blood pressure. These can all go the other way too. So you can have autonomic dysfunction where instead of being red, hot, sweaty, you can be cold, pale, and sleepy. And really it depends on what constellation of symptoms you're seeing to decide what is the right way to treat, because you would not want to treat someone whose primary dysautonomia symptoms are sleepy, cold, and tired, or with something that would actually dial down autonomic output. You'd actually want to use something that actually wakes them up. So it really just depends on what phenomenology each individual patient has. To follow up with that, you can have both. So some patients with autonomic dysregulation can swing wildly from really hot, agitated to really slow and cold, sometimes with even the same day.
Okay, thank you. And this is a question for Heather. How important is the syndrome versus the genetic cause?
That's a good question. So the syndrome and the seizure types and the EEG patterns are helpful with very much helpful in deciding which medications you're going to use and helping predict outcome. The genetic information additionally gives information there because within a given syndrome, the genetic causes can have different predicted outcomes and also different treatment approaches. So with SCN2A, for example, if it's a known gain-of-function mutation or suspected gain-of-function mutation, you'd be more tempted to try Dilantin or other sodium channel-acting medications that have been shown to be particularly beneficial compared to another genetic etiology. So having a genetic diagnosis early can certainly help in making those decisions. Your seizure doctor is definitely going to pay attention to the syndrome and the seizure and EEG features first and foremost, but the genetics can add another layer of information to be helpful, and we hope more and more helpful over time, especially as clinical trials come out and you'll need a genetic diagnosis to qualify if there's something more targeted.
Okay, and one additional question for you, Dr. Olson. How can cortical visual impairment be diagnosed?
Great question. So it's a clinical diagnosis, and in the most ideal setting, it's actually a combination of a neurologist, an ophthalmologist, and a functional visual assessment, which is usually done by a teacher for the visually impaired. We don't often get all three of those pieces, but having a high level of suspicion and working with your neurologist and or ophthalmologist to say, is this an issue for my child? You would suspect it if your child is having trouble with their vision or has some variability in their visual attention. So sometimes they're able to look at you and follow very well, and other times they're just not as engaged, separate from seizures, for example. So if you're at all worried about the vision, it's something I would say definitely ask about. And there's a lot of variability in knowledge about CVI, so seeing just any ophthalmologist may or may not get you the answer that you need because the training on CVI is actually fairly minimal in ophthalmology training and in neurology training. So finding somebody who knows about cortical visual impairment and can help assess your child would be the best approach.
Okay, and this is a question for Dr. Coffman. Have you noticed more dystonia or dysautonomia symptoms in patients with uncontrolled seizures or changes in anti-epileptic medication levels, and appropriate treatment regimens can limit these symptoms?
So the short answer is yes. With epilepsy that is not well controlled, dystonia, dysautonomia are often much more pronounced. So when my complicated patients have flares of their dystonia, dysautonomia, one of the questions that I ask is, how are we doing from an epilepsy control standpoint? Because if the seizures are well controlled, then I know I can work on controlling the dysautonomia or the dystonia. But if the epilepsy has fallen out of good control, until that's improved, there's frequently nothing that I can do because the basal ganglia are downstream from the cortex to be able to get the dystonia or dysautonomia under control until the epilepsy is better controlled.
Okay, and another question, can adding iron help with movement disorders?
So the only movement disorder, it's not technically a movement disorder, that improves with iron therapy is restless leg syndrome. So this is a phenomenon with kicky, twitchy movements during sleep, very restless sleep, and that is the only movement disorder of which I'm aware that improves with iron supplementation. And the key critical thing is there is such a thing as too much iron. Too much iron in the body can actually be toxic to the liver. So it would not be anything to do without consulting your physician first.
Okay, thank you. And we do have some more questions regarding that, but I'm just going to jump back to some of the questions that we had from earlier presentations that we weren't able to address. So Dr. Chung, I believe that this was during your presentation, but somebody wanted to know if you had matched seizure control to any medications taken. Sorry, Dr. Chung, are you still on the line? Okay, so we'll just go on to a different question and we'll have Dr. Chung answer that question later. Dr. George, I think this question came up during your presentation, which is the CNVs play a large role in many epilepsy. CNVs play a large role in many epilepsy and autism. Will any functional studies include them?
That's a great question, and so the answer is no, but here's the why. Our expression system is primarily good at testing the effects of amino acid substitutions, and those result from changes typically in the coding region of the gene. CNV, which stands for copy number variation, often refers to much larger pieces of DNA that vary in their presence or absence in other regions other than right in the gene, in the protein coding part of the gene. So we don't have a method to determine the functional consequence of a CNV at this time.
Okay, and then there's a translational research question. Does anyone have a guess as to whether or not gene therapy or viral vector gene editing technology would help children who are already several years into their disease? It seems like we have the technology for replacing SCN2A gene. Is there a reason to think this would be curative or even helpful? And I think Dr. Sanders touched on this a little bit, but perhaps some of the other speakers on the line might be able to weigh in.
Yeah, this is Al George. I think Stephan Sanders did touch on this quite eloquently, and I think the answer is we don't really know. I think the likelihood that introducing a normal copy of SCN2A long after the seizure onset, when other things have already begun to happen as a consequence of that primary genetic insult, I think the likelihood that that will have a dramatic therapeutic benefit is less likely than if one were to start that kind of therapy early on. But this is really uncharted water, and we don't really know. It's going to be really valuable to have really well-validated animal models with prototypic mutations that we can test. Can we introduce the gene effectively at throughout the lifespan, and when does it have the most efficacy? I think these are really unsolved questions right now. It's an excellent question. I don't think we have the answers yet. But that doesn't mean people aren't motivated to try. I think it's just we're not there yet.
Okay, thank you so much. Another question is, are the SCN2A children more vulnerable at puberty? Should it be delayed? And I know this wasn't specifically one of our topics, but if any speakers on the line have any insight.
I can jump in on that a little bit. Puberty can be a vulnerable time for epilepsy, so in some patients that can be a challenging period and others less so. And some patients, actually, the seizures get better into adolescence. So it's a little bit variable. In general, we don't suggest delaying puberty. That would be perhaps in unique but unusual situations.
Okay. And then the next question is it possible to obtain a personalized therapy based on the type of mutation?
So this is Al George again. I'm happy to tackle that. This is an issue we think a lot about, and others in the field are also thinking a lot about this. I think our goal is to answer that question. Is there a medication? Let's, for example, talk about a sodium channel blocker. Phenytoin, carbamazepine, lamotrigine. There's half a dozen widely used sodium channel blockers available on the market. We do have a good sense now, and again, Stephan Sanders touched on this, that there's a dichotomy of functional consequences in SCN2A, where there are cases of autism and intellectual disability where there's very clear evidence that loss or reduced function of SCN2A is responsible or partly responsible. In those cases where there's already reduced function or absent function of the gene product, you probably don't benefit from having additional pharmacological block of other sodium channels. Whereas those mutations that are associated with very early onset seizures and severe cases of epileptic encephalopathy that really begin very, very early in life, that where the mutation has been shown to cause a gain of sodium channel activity, gain of function, the therapeutic goal there is to maybe to knock down the effect of the mutation and inhibit the sodium channel, attenuate the activity. And so there's sort of the broad brushstroke. And he referred to a paper, a multi-authored paper that published last year, first author being Wolf, where clinically it looks like those individuals who have very early onset seizures respond best to sodium channel blockers, and those that have later onset and maybe have more of an intellectual disability plus seizures phenotype or autism spectrum disorder plus seizures phenotype, they don't respond as well. Those are broad generalizations. What we don't know, and what I think is going to be possible to figure out soon, is whether individual mutations in SCN2A that are gain of function, but by different gain of function mechanisms, whether they're treated more effectively by certain sodium channel blockers. Maybe some mutations will be fixed really readily by phenytoin or carbamazepine, whereas others might require a different sodium channel blocker. And there are experimental sodium channel blockers being developed now that may come into play at some point in the future. So how do we understand all that? And our again, this is just a kind of self-promotion here, but we're thinking that we want to test that hypothesis in the simplest possible system where we only have the channel and minimal other cellular components. And there we can determine, is it a gain of function or a loss of function? And if it is a gain of function, how does that channel respond to a wide panel of sodium channel blocking drugs, some that are on the market for treating epilepsy, some that might be on the market for treating other disorders, some maybe not on the market yet? And can we then start to profile individually each mutation against this panel? And if we begin to see some heterogeneity in the responses, then I think the answer is going to be that, yep, there may be a personalized approach here, that every mutation needs to have a pharmacological fingerprint before decisions about treatment are solidified. But having said that, easier said than done. It's a challenge to do it fast enough that a child being treated, that the decision can be made to switch drugs early enough that it matters. So that's it was an excellent question, and I hope one day we can answer it.
Thank you so much. The next question is, what factors limit genetic testing in ASD later onset epilepsy population?
Sorry, can you repeat that?
Sorry, I'm just It's what factors limit genetic testing in ASD? And I think it's in ASD.
It's really just insurance approval, as well as having programs that are focused on that. But insurance has been the biggest factor, at least at our center.
Okay, thank you. And this is a question about drug recommendations. What drugs do you recommend for children with both SCN1A and SCN2A gene deletion?
So with the deletion syndromes that involve both genes as well as sometimes some other sodium channel genes, usually the SCN1A is the biggest driver, so they end up being more on the order of Dravet syndrome in that spectrum, and so the medications that are recommended in that subgroup, but it does need to be relatively individualized.
Okay, and then this question is...
I'll just add to that. Sorry, that usually with a loss of it, you'd have loss of SCN2A, and it'd be a loss of function, so you would still, in general, avoid sodium channel blockers like you would with the Dravet kits.
Thank you. So this next question is, if a child has more dystonia, movements, and pain as they have fewer seizures, would you know why that happens?
Yeah, so this is actually a fairly common phenomenon within the epileptic encephalopathies, that when the cortex gets under better control, meaning the epilepsy gets under better control, the basal ganglia have been primed, if you will, by all of the hyperactive signals going to them. So they start to fire independent of what's going on at the cortex, and you get worsening of movement disorder because the basal ganglia are reacting to the input that they used to have and firing abnormally. So it's very frequent for me to see patients from my epilepsy colleagues that now their epilepsy is under good control. They've had either the new development of or the severe worsening of a movement disorder because of that phenomenon.
And another question that we got is, do you suggest to apply cannabinoid therapy? Do you have any comments on that?
And from a movement disorder standpoint, movement disorder standpoint, there is no data regarding the treatment of hyperkinetic movement disorders with cannabinoids.
From an epilepsy perspective, that's a complex question that deserves a whole conference in and of itself. Epidiolex is something that is coming out in the near term and has been approved and studied in specific disorders, including Dravet, but not in SCN2A specifically, and a lot still needs to be determined there. So it's not something that I necessarily advocate broadly, but it's situation dependent.
Okay, and this question is just asking for the speaker's opinions on triliptol and its effectiveness. And I may have mispronounced that, I apologize.
Is triliptol what you said?
It's, sorry.
Yeah, that's exactly what she was trying to pronounce, triliptol.
Okay, so triliptol or carbazepine is the other name, is one of the anti-seizure medications that is sodium channel blocking. And it's for kids with gain-of-function variants, it's amongst the group of medications that we think may be beneficial, but there isn't enough data on that one medication in and of itself
to say definitively. But it's a good medication for focal seizures.
Okay, thank you. And sorry again about the mispronunciation. So the final question that we have here is just asking if the speaker could repeat about the recommendation of supplementation for spasms, and is it valid for epileptic spasms?
Supplementation for spasms? Sorry, I didn't quite understand the question.
I'm not sure which speaker the question was for, but it just says, could you please repeat about the recommendation of supplementation for spasms? Is it valid for epileptic spasms?
This is Dr. Olson. I'm not sure if that question is targeted for me or not. I talked about the spectrum of seizures, and I did mention epileptic spasms, but I don't recall mentioning anything about supplementation specifically. So we may have to clarify. I'm happy to answer questions by email.
Okay, all right. Thank you so much. Well, that's all of the questions that we have. So I'm going to go ahead and let Jennifer Chernago from Simon's VIP just do a little bit of a recap.
Hi, great. So I just want to start by just thanking all of the families that have participated in providing information to all the researchers, all the data that you've already entered into Simon's VIP registry and continue to enter. And also, definitely kudos to the FamilieSCN2A Foundation for working hard to identify and various research groups. There's an amazing group of collaborating organizations that many are represented here, and I think it's really going to make a difference. And just to echo what Dr. Sanders said earlier on, that the data that you're providing really is making an impact in real time. Actually, Dr. Chung and I were out at the Global Genes Patient Advocacy Research Summit this week, and heard over and over again about how important it is to have a well-developed patient registry with standardized and good quality data when you get to the point of wanting to submit for clinical trials and submit for approval through the FDA. So again, thank you for that. To just reiterate also the data that we have available in Simon's VIP for researchers. We did have a new data release last month. And as we said, there's data available for 58 individuals with SCN2A, and there's going to be more to come. For biospecimens available, you can see the numbers there, but I'll also say for all the families on the line that we have a new contract with Quest Diagnostics, which usually are local to your locations. So we have increased capacity at this point to, in a remote fashion, get blood draw samples from you by sending you kits, and you go to your local Quest center. So we're hoping that will continue to increase the availability of biospecimens to external researchers all over the world. And for any of the researchers online that are interested in learning more about accessing these resources, the website is there. Next slide. And for just to say again where we are currently, I think Dr. Chung had a slide on this. We have 114 individuals registered currently, which is an amazing, amazing amount of response. And then you could see over time how that 114 registered translated to 86 that were consented with and had their lab reports uploaded, and then a lower number then had completed their medical history. So as you see, these are the various steps for participating in the VIP project. Some of these are online, where you add your children to the account, you complete your consent forms so that you understand what the research is all about. You answer some questionnaires and upload your genetic report, and then you do this phone call with a genetic counselor to get in-depth medical history information and some other instruments collected at that point. And then there are additional online research surveys. So if you find that you are not quite clear on where you are in the process and want to double check that you've done all the steps that are in this flow here, there's an email on the slide below. You can feel free to reach out to our coordinators and they can help you at any time figure out if you're all done with what you need to do right now, or if you need to have a few more steps completed. So again, I just want to thank all the families and everyone that's been contributing so far, and we're very excited to continue to grow this group.
All right. Thank you so much, Jennifer. And now I'll let Carla from FamilieSCN2A Foundation say some closing remarks.
Hello, everyone. My name is Carla Forbes. I am the co-founder and vice president of the FamilieSCN2A Foundation. On behalf of the foundation, we would like to thank our presenters for taking the time to share updates in SCN2A research. Also, thank you to the Simons VIP Connect team for co-hosting our conference and for their continued support with Families SCN2A. The entire SCN2A community is thankful for your drive and dedication for all SCN2A disorders. We hope everyone listening today realizes that you are an important piece in this journey. Each one of us can create a ripple to form the ultimate wave. Spreading SCN2A awareness, registering to the central database, donating, advocating for change, be a volunteer with the foundation are examples of ripples in need. So we ask you today, how will you create your ripple to support the wave? Our community needs research, and researchers need the community to continue the wave to cure SCN2A. We thank you for joining today, and we hope to hear from you soon.